me, please inform me to start. Okay, very good. So in the name of God, in order to continue our lecture, we are on the page 64, patterns of transmission. We are talking regarding to the patterns of transmission. As this slide shows, there are two important ways for transmission are present. One is direct contact. Direct contact, okay? Like physical contact, droplet nuclei, and aerosols. It is droplets by coughing, sneezing. Many microbes can go out from respiratory tract of one infected person and transmit it to another sensitive one. So aerosol, droplets, physical contact, yes. Here you can see person to person, airborne by hand or object contact, by respiratory droplets, and by contact with animals, it is indirectly. So indirect contact passes from infected host to intermediate conveyor and then to another host. And vehicle, there are many vehicles are present for indirect transmission: food, water, biological products, and vomits, as you see, for example, by contaminated food, by contaminated water, by contact with contaminated object, and the most probable object is contact lens, which some people use on their eyes if it is infected, because eyes are very sensitive, so it can be a transmission device. By injection of contaminated soil, by arthropod bite, yes, any insect like sandfly, tesserae, and anophel, many types of arthropods, insects, and some like mites, fleas, can be responsible for transfer of microbes from one person to another person. So food, water, biological products, and vomits. Because COVID-19 in these days is very important, so I put one slide related to coronavirus. Transmission of COVID-19, human atomization of virus arise from coughing or sneezing of an infected person, producing virus containing droplets. If these things coming out from coughing and sneezing are sizes are more than 5 microgram, we call it droplets. If the size is less than 5 micrometer, we call it aerosols. So aerosols and droplets. Aerosols are mostly remaining in the air and transmitting to inhalation of another person, but droplets mostly coming down to contact to any devices and then by contact. So another person get hand or any contact, it can be transmitted to new person. So human atomization of virus arise from coughing or sneezing of an infected person, producing virus containing droplets more than 5 microgram or aerosols less than 5 micrometer. Virus transmission from person to person occurs through direct, indirect contact, and airborne aerosol droplet routes. Large droplets, this is large droplets, mainly settle out of air to cause person-object contamination. Person-object contamination. While aerosols are efficiently dispersed in air, direct and airborne transmission occur in short range and extend distance time, respectively. Inhaled airborne viruses deposit directly into the human respiration tract. So you can see by aerosols can directly for a short time transfer from one person to another person, or by droplets they come to different devices and then by contact, indirect contact can transfer to a sensitive person. Here there are two parts in this figure: modes of infection, disease transmission. Modes of infectious disease transmission. General transmission. So you can see abiotic environmental factors. It can be transmitted by abiotic environmental factors like wind, water, inhalation of spores, and entry into a skin from one person to another person, or by animal vectors like mosquitoes, like malaria, dengue, and fleas like bubonic plague. So in this figure, we were to understand modes of infectious disease transmission. In part A, pathogens can be transferred by environmental factors such as wind, water. They can also be transferred between humans, as well as from humans to animal vectors. Animal vectors can further spread the disease through migration if carried by birds or fish or trade. In second part, human to human transmission. We want to see human to human transmission. One is direct contact. Direct contact. Pathogen survive best inside the body, like HIV, herpes viruses, Ebola, and mostly they cannot survive outside the body. So directly from one person to another person in a short time, transmission is happening. Indirect contact. Pathogen survives harsh environment. They are not sensitive as in group 1. In group 2, they can be on different devices. Pick up pathogen from surface or air like influenza, norovirus, droplets. Pathogens are in droplets but don't survive long this way, like Ebola, Bordetella pertussis. Airborne pathogens aerosolized and stay infective. So immediately by air, transferring from one person to another person. Influenza, tuberculosis, fecal, oral, through contaminated water or food. So those pathogens are present in the feces of the patient. So feces can contaminate water or food or vegetables. And the other sensitive people, by eating those food, water, or vegetables, get infection also, like chlorella, norovirus, shigella, and etc. So according to the figure, human to human transmission has been classified into 5 main modes. These 5 modes are not mutually exclusive. For example, the Ebola virus can be spread through direct contact and potentially through droplet transmission. So if we have one pathogen, sometimes it is not only one way for transmission. Sometimes by 2 or 3 ways they can reach from infected person to sensitive one. How pathogens can be transmitted mostly depends on how hardly they are outside a human body, how much they can suffer outside the body, how long they can be alive outside the body. So this resistance shows the ways they can use for transmission. Some cannot survive for long periods of time, so they require direct contact, droplet transmission, because in droplet around pathogens there are lots of water. So water help them to survive the adverse conditions of the environment, or transmission through animal vectors. So when they are sensitive outside the body of humans, some of the pathogen going through the body of animals or insect, and accordingly in a suitable time they reach to the body of another sensitive human person. Others, such as flu, can survive for long periods of time on surfaces, making them extremely contagious. Fecal oral pathogens are a large problem in developing countries, because in developing countries, sometimes using the feces of human as fertilizer for agriculture. So those agriculture people make contaminated vegetables with many pathogens. And during working and during farming, those pathogens can reach to the bottled water and wells and rivers and make them contaminated also. Okay, 68. Transmission. Because in some pages that are very simple and easy to understand, I'm going more fast than before. But if you find any problem, you can stop it and asking your question. So when it is very simple, there is no need for long time explanation. And we have a plan to finish our lecture today, all the lectures. So next week, according to the Iran calendar, is holiday. So next week on 5th June, we don't have lecture. But after 2 weeks, at 12 June, we will be in the class to answer the questions and reviewing and talking about examination. So don't forget, next week there is no lecture, but after 2 weeks at 12 June, okay, at 12 June, I think it is 12 Zilqa'dah also. We have another class, but not for teaching any new thing, for talking about examination, answering the question, and review. And today we are going fast to finish the remaining pages. Transmission. Transmission must occur for an infection to spread. So if one community wants to be infected, infection should be transmitted from person to person. First, transmission from the reservoir to the individual must occur. In some cases, as we explained, some bacteria are sensitive, so they found reservoirs. Some reservoirs are animals, and some reservoirs are insects and arthropods. So if any infection have reservoir, transmission from the reservoir to the individual must occur first. Then the individual must transmit the infectious agent to the other susceptible individuals, either directly or indirectly. Pathogenic microorganisms employ diverse transmission mechanisms. Contact transmission. Contact transmission. It includes direct contact or indirect contact. Person-to-person transmission is a form of direct contact transmission. Here, the agent is transmitted by physical contact between two individuals through actions such as touching, kissing, sexual intercourse, or droplet sprays. Part 1. Direct contact can be categorized as vertical, horizontal, or droplet transmission. First, we are to go on vertical transmission. What is vertical transmission? Vertical direct contact transmission. It occurs when from mother to a child during pregnancy, birth, or breastfeeding. In any time when a fetus or a newborn has a very close relationship to mother, maybe any infection, if present in the mother, can be transmitted to the fetus and newborn during pregnancy, during birth, or during the breastfeeding. It is vertical direct contact transmission. There are many pathogens can have vertical transmission. Some of them are viruses and some are bacteria. Some viruses like Zika virus, chickenpox, hepatitis C, D, E, enterovirus, AIDS, HIV, parvovirus, and rubella, cytomegalovirus, herpes simplex, papillomavirus, and some are protozoa like Toxoplasma gondii. But because our course is advanced bacteriology, so mostly we focus on some bacteria. In all others, like group B, Streptococcus, Listeria, Streptococcus, and yeast like Candida, Lyme disease. E, everything else sexually transmitted, gonorrhea, chlamydia infection, uraplasma, urolithicum, and syphilis are those bacterial pathogens which I showed by these arrows. If any microbe can be transmitted from mother to fetus, we call it congenital transmission. From mother to fetus by placenta, we call it congenitally transmitted infection. So if during pregnancy one mother has one of these infections, these pathogens can be transmitted vertically from mother to the fetus. Another transmission is horizontal direct contact transmission. Horizontal direct contact transmission. Okay, dear student Jawad Ajmil, I told that in next time we only talking about the examination. So all of your questions you can ask me in the next class. If now we talk about the examination, some of the today's lecture remaining to explain for next time. Okay, so today only is the time for finish the lectures, and next time it is the time for talking about examination. Okay, thank you. Horizontal direct contact transmission. Often a contact between mucous membrane is required for entry of the pathogen into the new host. Although skin-to-skin contact can lead to mucous membrane contact, if the new host subsequently touches a mucous membrane, it also be site-specific. For example, some diseases can be transmitted by sexual contact, of contact. And the third one in this part is droplet transmission. Droplet transmission. When an individual coughs or sneezes, small droplets of mucus that may contain pathogens are ejected. This leads to direct droplet transmission, which refers to the droplet transmission of a pathogen to a new host over distances of S. A wide variety of diseases are transmitted by droplets, including influenza and many forms of pneumonia. Transmission over distances greater than 1 meter is called airborne transmission. Airborne transmission. So we have transmission By air less than 1 meter, it is droplet transmission, and transmission in distances over than 1 meter, greater than 1 meter, it is called airborne. So it is here you could understand the difference between droplet transmission and airborne transmission. In some cases are the same, and in some cases they are different. Okay, right now we are in the page 71. 71. Indirect contact transmission. It involves inanimate object called fomites that become contaminated by the pathogens from an infected individual or reservoir. For example, an individual with the common cold may sneeze, coughing, droplets to land on a fomit, such as tablecloth, carpet, or individual may wipe her nose and then transfer mucus to a fomit, such as a doorknob or towel. Transmission occurs indirectly when a new susceptible host later touches the fomit and transfers the contaminated material to a susceptible portal of entry, like after using hand for opening the door, the doorknob which is contaminated, then by finger touch, nose touch, eyes, or eating food by those contaminated hands, and indirectly those bacteria can transmit it to the person. Fomits can also include objects used in clinical setting that are not properly sterilized, such as syringes, needles, catheters, surgical equipment. Pathogens transmitted indirectly via such fomits are a major cause of healthcare associated infections. Here in this figure, we can see fomits are ennobling objects that facilitate the indirect transmission of pathogens. Contaminated doorknobs, towels, syringe are common examples of fomits. 73. Vehicle transmission. In the next part, we are going to talk about vehicle transmission. The term vehicle transmission refers to the transmission of pathogens through vehicles, such as water, food, and air. Water contamination through poor situation methods leads to waterborne transmission of disease. If any infection can be transferred from source of infection to a new sensitized, sensitive person, we call it waterborne. If it is transferred by food, we call it foodborne. If it is transferring by air, we call it airborne infection or airborne transmission. So we have waterborne, foodborne, airborne transmission. Water contamination through poor sanitation methods leads to waterborne transmission of disease. Waterborne disease remains a serious problem in many regions throughout the world. The World Health Organization, WHO, estimates that contaminated drinking water is responsible for more than 500,000 deaths each year in the world. Similarly, food contaminated through poor handling or storage can lead to foodborne transmission of disease. Dust and fine particles known as aerosols, which can float in the air, can carry pathogens and facilitate the airborne transmission of disease. For example, dust particles are the dominant mode of transmission of hantavirus to humans. Hantavirus is found in mouse feces, urine, and saliva, but when these substances dry, they can disintegrate into fine particles that can become airborne when disturbed. Inhalation of these particles can lead to a serious and sometimes fatal respiratory infections. 74. Although droplet transmission over short distance is considered contact transmission, as discussed above, longer distance transmission of droplets through the air is considered as vehicle transmission. So transmission over short distance, we call it contact transmission. For a longer distance, we call as vehicle transmission. Unlike larger particles that drop quickly out of the air column, fine mucus droplets produced by coughs or sneeze can remain suspended for long periods of time, traveling considerable distances. In certain conditions, droplets desiccate quickly to produce a droplet nucleus that is capable of transmitting pathogens. Air temperature and humidity can have an impact on effectiveness of airborne transmission. Example is tuberculosis. Tuberculosis is often transmitted via airborne transmission. When the causative agent Mycobacterium tuberculosis is released in small particles with coughs. Because tuberculosis requires as few as 10 microbes to initiate a new infection, patients with tuberculosis must be treated in rooms equipped with special ventilation, and anyone entering the room should wear a mask. Another one is vector transmission. Vector transmission. Disease can also be transmitted by mechanical or biological vector. An animal, typically an arthropod, that carries the disease from one host to another. So it is vector. We have two types of vector: mechanical vector and biological vector. What is the difference between mechanical vector and biological vector? When one infection, when one microbe has different ways for transmission, and one of the ways is vector. It is mechanical vector. And on the other hand, without any changing, only resistant inactive form of the pathogen is transferred by mechanical vector. And if this vector is not present, pathogen have possibility to transmit it by another means, by another ways. But in biological vectors, in biological transmission, the vector is the only way of transmission. The vector is the only way of transmission. And some parts of life cycle of the pathogen is done in this vector. So the presence of this vector in the life cycle of pathogen is obligatory. It is necessary. Without this vector, it is not possible for completion the life cycle. So mechanical transmission is facilitated. It is not necessary, but if it is there, it is good. If it is not, another ways is facilitated by mechanical vector. An animal that carries a pathogen from one host to another without being infected itself. So the vector is not infected. Only transmission is happened. For example, a fly may land on fecal matter and later transmit bacteria from the feces to food that it lands on. A human eating the food may then become infected by the bacteria, resulting in a case of diarrhea or dysentery. Here you can see, yes, for example, there is a fly on feces, human feces. Fly picks up pathogen from fecal matter and carries it on its body. Number 2, fly transfers pathogen to food. Now fly is there and pathogen is on the food. Number 3, person eats contaminated food and then get sick. So by different ways, this bacteria can come to food or vegetables or water, and this fly is not the only way. And this fly is not infected, only carrier. This fly doesn't have any problem. This fly is not sick because of the presence of the pathogen. No, this mechanical, this fly which carry the bacteria cannot be the biological vector. One fly is, the presence of one fly is obligatory or not obligatory. If the presence of a fly or an arthropod is obligatory and for completion of the life cycle, its presence is obligatory, it is biological vector. And if it is not obligatory, it is mechanical. It is not possible, some cases to be biological and some cases to be mechanical. A mechanical vector carries a pathogen on its body. Yes, on its body, not inside its body. A mechanical vector carries a pathogen on its body from one host to another, not as an infection. But in the B, you can see a biological vector carries a pathogen from one host to another after becoming infected. For example, you can see over here, this fly is contaminated, is infected. So infected mosquito bites an infected person. So by biting, pathogens coming to the body of the person. Okay, like for example, leishmania. Then infection spreads through the body and into the red blood cells, like malaria, for example. First going to the liver and second going to the blood. Now, this mosquito is without any pathogen, coming only for eating or drinking blood. So when this person is infected, those pathogens goes through the alimentary canal of the insect. Secondary mosquito bites infected person. Mosquito may now transmit infection to the another person. And some parts of the life of pathogen is completed into the body of biological vector. Some parts of the life of pathogen completed in the body of this mosquito. So this mosquito is infected, but this fly is not infected. It is the difference between mechanical and biological vectors. So in this example, there we explain that this fly transfer pathogen to the blood. In this mechanical vector, we explain that those flies sitting on feces and pathogens are on, and when they sit on food or water or vegetables, contaminated. Never we explain that by transferring to blood. Transferring to blood not happened. It is transferring of pathogen to food or vegetables and then eating, and pathogens going to the alimentary canal of the person, not going to the blood. Here there is no contact of insect to human. There is no transmission from insect to the body of human. It is transmission from insect to food. And then food is the causative agent for starting infection. Yeah, the second example for biological vector is can be have example like malaria, like sleeping sickness, like American trypanosomiasis, and many others, which the presence of fly is compulsory, is obligatory. Okay, there is no problem. Mr. Ali Abdul Qadir Saadi, there is no problem, but what can we do? They should try to come to the lecture. So their absence, no problem. And after some time, again, you can hear the lecture, recorded lectures, and if they have questions, you can ask me. Their absence is not problem according my side. Biological transmission. It occurs when the pathogen reproduces within a biological vector that transmits the pathogen from one host to another. Arthropods are the main vectors responsible for biological transmission. Most arthropod vectors transmit the pathogen by biting the host, creating wound that serves as a portal of entry. The pathogen may go through part of its reproductive cycle in the gut or salivary gland of the arthropod to facilitate its transmission through the bite. For example, hemipterans called kissing bug or Assasian bug transmit Chagas disease to humans by defecating when they bite, after which the human scratches or rubs the infected feces into the mucous membrane or bricks in the skin. Biological insect vectors include mosquitoes, which transmit malaria and other diseases, and lice, which transmitted typhus. Other arthropod vectors can include arachnids, primarily ticks, which transmit Lyme disease and other diseases, and mites, which transmit scrub typhus and rickettsial pox. Biological transmission, because it involves survival and reproduction within a parasitized vector, complicates the biology of the pathogen and its transmission. Complicating. So vector complicate the biology of the pathogen and its transmit, not only complicated, but also completed. So the presence of biological vector is necessary, is compulsory, is obligatory, is needed for completion the life cycle of the pathogen, but it is not so for mechanical vectors. There are also important non-arthropod vectors of disease, including mammals and birds. Now we want to see how one mammal can infect us, how one bird can infect us. Various species of mammals can transmit rabies. It is a viral infection to human, usually by means of a bite that transmits the rabies virus. So dog. So this virus is present in some cases of dogs. And when dog biting a person, this virus is transferred from dog to human and sometimes cats or similar animals. Chickens and other domestic poultry can transmit avian influenza to human through direct or indirect contact with avian influenza virus? A. Shed in bird saliva, mucus, and feces. Okay, the other part, the last part remaining to finish our lecture today is talking regarding epidemiology. Epidemiology. What is that? Wherever we see logy, like bacteriology, immunology, microbiology, logy means study. Study of epidemics. And what is that? Study of the frequency and distribution of disease and health-related factors in human populations. We call it epidemiology. So, when pathogens are our enemy, and when these enemies attack not only to one person, but also to different groups, different populations, in one country and sometimes different country, it is necessary we know their epidemiology. So when we want to defend ourselves, against any pathogen, we should know their structure, we should know their biology, we should know their metabolism, we should know their resistance and sensitivity to drugs and vaccines, and we should know the current epidemiology, in which part of the city it is more, in which parts of the country they are more, in which parts of our neighbors and other countries and other populations they are more, and then programming for defend. Okay, before doing anything we should have knowledge. So epidemiology is very important for programming to stand against our pathogens. Surveillance, collecting, analyzing, and reporting data on rates of occurrence, mortality, morbidity, and transmission of infections. It is surveillance. Reportable, notifiable disease must be reported to authorities. So some people are present in any hospital, in any city, in any capital, and many places in the world which they should see the data and then programming for defense. Okay? For example, in the United States, Centers for Disease Control and Prevention, CDC, is present in Atlanta. So in all the other countries also, there are similar places like CDC are present, which they do programming for defense against these enemies, these small tiny enemies. Principal government agency responsible for keeping tracking, minute to minute, hour to hour, day to day tracking of infectious disease nationwide. What is prevalence? What is prevalence? Total number of existing cases with respect to entire population, usually represented by percentage of the population. So, prevalence is dividing total number of cases in a population, infectious disease, to total number of persons in the population, multiply 100. So whatever we find, it is the prevalence. The prevalence. And when we talk about disease, because we are microbiologists, mostly we are focusing on infectious disease. So we have two types of disease. Some diseases are non-infectious and some diseases are infectious. Infectious diseases are those disorders that the causative COVID-19, Mycobacterium tuberculosis, and many microbes which can work as a pathogen. Non-infectious disease like diabetes, like, for example, migraine, anemia, and many abdominal problems, gastrointestinal problems not related to pathogen. They are not non-infectious diseases. So here, when we want the prevalence, we want to know the prevalence of infection, the total number of cases in population. Cases of what? Infected people. In total number of persons in the population. Sometimes we can understand it for example, the prevalence in one village, the prevalence in one city, the prevalence in one area, or the prevalence in a country or many countries or whole world. So we should first thinking which area we want to know the prevalence, and accordingly we found the cases and dividing to the total number of persons on that place. Incidence measures the number of new cases, not all cases, new cases over a certain time. We should have a time, for example, during 6 hours, during last day, during last week. So a certain time we focus and then we measure the numbers of new cases over a certain time period as compared with the general healthy population incidence, the number of new cases divided to number of healthy persons, and it is a ratio we can have it to compare with another places, another regions, another countries. And you can see incidence and prevalence are not completely matched to each other. They can go their ways during the different years. It is the incidence and prevalence of HIV or AIDS. Mortality rate, total number of deaths in a population due to a certain disease. It is mortality rate. But what is morbidity? number of people afflicted with a certain disease, we call it as morbidity. So the death, mortality. People afflicted, morbidity. Sometimes one infectious disease all the times present in one place during 1 year, 5 years, 10 years. Whenever we check the time in the previous time, they are present. Or in another case, endemic is a disease that exhibits a relatively steady frequency over a long period of time in a particular geographic locale. We don't mean high or low. If one infection, high or low, no difference, to be for a long time in one place, that infection is endemic on that place. Sometimes it can be a part of a country, sometimes it can be a whole of a country, or some different countries. This is that exhibits a relatively steady frequency over a long period of time in a particular geographic locale. Sporadic, when occasional cases are reported at irregular intervals. So, for example, some infections are present in one place, then it is not there. After some time, another place. It is sporadic. Sporadic. For example, now monkeypox. In these days, we hear it is sporadic. A few is in England, a few is in Poland, a few is in another country, few. It is sporadic. Epidemic, when prevalence of a disease is increasing beyond what is expected. So, when the prevalence of one infection is not irregular, it is increasing beyond what is expected. So it is epidemic. Epidemic. And if it is in one country, we call it epidemic. And if it is across many countries or whole world, we call it pandemic. Pandemic. Quarantining. Quarantining. Individuals suspected or known to have been exposed to certain contagious pathogens may be quarantined or isolated to prevent transmission of the disease to the others. Hospitals and other healthcare facilities generally set up special wards to isolate patients with particularly hazardous disease, such as tuberculosis and Ebola. Yes, COVID-19 now is pandemic, yes, of course. Depending on the setting, these wards may be equipped with special air handling methods, and personnel may implement special protocols to limit the risk of transmission, such as personal protective equipment or the use of chemical disinfectant sprays upon entry and exit of medical personnel. The duration of quarantine depends on factors such as the incubation period of the disease and the evidence suggestive of an infectious. The patient may be released if signs and symptoms fail to materialize when expected, or if preventive treatment can be administered in order to limit the risk of transmission. If the infection is confirmed, the patient may be compelled to remain in isolation until the disease is no longer considered contagious. So sometimes it is needed the patient to transport from one hospital to another hospital, or from one city to another city, or from one country to another country, and the patient have a serious contagious infection. So here you can see in figure A the aeromedical biological contaminant system, ABCS. It is an ABCS, aeromedical biological contaminant system, is a module designed by the CDC and Department of Defense especially for transport highly contagious patients by air. So when you want to transfer these patients by airplane, first they should quarantine in this equipment and not allowing the other personnel or other people infected by this pathogen. B, an isolation ward for Ebola patients in Lagos, Nigeria. You can see over here. So it is a hall in a hospital with limited going and come back. Only personnel can going there with equipped mask and cover. And the infected people are hospitalized in such a place. An isolation ward for Ebola patients in Lagos, Nigeria. In the United States, public health authorities may only quarantine patients for certain diseases such as chlorella, diphtheria, and infectious tuberculosis, and strains of influenza capable of causing a pandemic. Individuals entering the United States or moving between states may be quarantined by the CDC if they are suspected of having been exposed to one of these diseases. Although the CDC routinely monitors entry points of the United States for crew or passengers displaying illness, quarantine is rarely implemented. Okay, so 88 means it is finished. So now we don't have anything to continue. It is sufficient. And after 2 weeks, as I told you, in the day 12 June, Sunday 12 June, we join to the class again and we talk about the examination, answering the question, and if needed, some review. If there is any question, you can ask me. If not, we can finish our course today.